All right. All right, I think we're ready.
Okay. All right, everyone, thanks for coming today. I'm really excited to introduce my friend Jessica Hekman so she can give her talk about canine behavioral genomics. She and I are friends from grad school, but she actually was a programmer before that and then got a veterinary degree at Tufts and then got a master's in comparative biomedical sciences to look at stress behaviors and how that relates to cortisol in hospitalized dogs. She also did a shelter medicine veterinary internship. And after that, she did her PhD at the University of Illinois at Urbana-Champaign, which is where we met. And since then, she's been working as a postdoc at the Broad Institute in the Carlson Lab, looking at canine behavioral genomics. And she's now a computational biologist there. So that's what she'll be talking about today. So I'm excited to give her the floor.
Thank you. Good to see you again, Kelsey. Yeah, so I'm going to talk basically, a lot of this talk is going to be about what Carlson Lab does and why we do it this way. And then at the end, I'll talk a bit more about exactly what I'm working on. And I'm happy to take questions about that. And the dog citizen science stuff is it's a large part of what Carlson Lab does, but it's only part, but it's what I'm focusing on for this talk because it's what I actually participate in and know about. So we do some other things like phylogenomics across hundreds of mammalian species, but I don't know anything about that. So, oh, and there's some tick research too. All right, so Kelsey covered that I'm a computational biologist. The Broad Institute is in Cambridge. For anyone who doesn't know, it is a human genomics institute, and we are the lone little lab that studies dogs, and we study dogs sort of as models for humans. And also just a little bit of background, you guys probably mostly know that so it's mostly, there's this perspective that dogs evolved from wolves. More recent research not done by our lab has suggested that in fact, they had a common ancestor and then there was a split and then you get wolves and dogs are sort of branched off, but they did not come from wolves. Wolves and dogs came from a third party. That split was somewhere between 15 and 40,000 years ago. There is a very active debate, a lot of research going on about when that happened. Was it before agriculture? Was it after agriculture? Why did it happen? How did it happen? Where did it happen? The only thing we're sure about is that it did not happen in the Americas, Australia, or Antarctica. But other than that, I think most everywhere else is pretty much up for grabs. So there's a lot of work going on there. So then once we had dogs and dogs were domesticated and living with humans, there was a lot of selection that started going on at that point, right? So it's at that point, it was like, oh, it's useful to have a dog that's big or a dog that's small. And that was when you started seeing genetic diversity decreasing a little bit in these different populations, which we have come to call breeds. I'm just going to move the thing here because I'm having trouble seeing my slide. Yeah, and so for all of that time, dogs have been adapted to live with us, which makes them really useful as models for studying human traits and human diseases because they live in the same places that we live. They share our environment, unlike a lot of other model species that we use, often like mice. And we do have a lot of these different types of dogs bred to do different jobs. And there's some debate about whether the dog that's in your house as your pet is really still able to do those jobs. But back when we had those breed splits about 150 to 200 years ago, we were selecting at the time on dogs who were able to herd or guard or run very fast or bring things back to you. And so one of the things that Carlson Lab does is look for genetic differences between these different groups of dogs. So what our hope is and our belief is that dogs are going to share not just the same traits, but some similar underlying, that the underlying biology and that the genes that control these traits are going to be similar in dogs and humans. And there's a lot of really interesting behavioral traits, aggression, anxiety, cognitive dysfunction, A lot of you maybe have dogs with storm phobia. So all of that is something that is really useful to look at dogs, use dogs as a model species for. And of course, there are a lot of diseases that dogs and humans also share. Dogs have been shown to be an excellent model for cancer. There are particularly some cancers that are much more common in dogs than are humans, and than humans, and therefore a lot easier to study in dogs than in humans. Carlson Lab focuses much more on the behavioral stuff, but we are actually starting to spread out into the disease stuff as well. We have a cancer project right now, which I'm not talking about in this talk because, again, it's not my project, but it's a very interesting one. And if anyone has questions about it, I may be able to answer some, and I certainly could point you to someone to talk to. So that was the idea, that we could take a look at changes in DNA, and differences in DNA between different populations of dogs, and that will help us understand dog behavior. And the idea is that once you understand the biology underlying a particular behavioral trait or a particular disease, that you can, I think most of you probably already know that this is the reason that we do behavioral genomics, is that if you understand the processes better, then you can hand that off to the people who actually do useful things, who can start studying how to fix the issues. And so the project name is Darwin's Ark, and I'll talk about that in a bit more detail later. And it is Carlson Lab is spread across UMass Medical School and the Broad, and then the International Association of Animal Behavior Consultants also participates in Darwin's Ark. So the story of Carlson Lab starts back 2005, the misty history of 2005, which was when the dog genome was first sequenced. This is obviously the cover of the Nature publication with that first paper, and it's such a fantastic cover because they have Watson and Crick, and they have photoshopped into the picture with them Tasha, who is the boxer who was sequenced, and then obviously they added a few more dogs, checking out how cool it is that we had the dog genome at that point. So that's Tasha. Well, most of you probably have a fairly good grasp of about how many base pairs are in human, 3.1 billion. There's 2.4 billion bases in dogs, so it's on the same order of magnitude and about the same number of genes. Right here, we have about 1,000 fewer genes, but I'm gonna say that I suspect that it's probably pretty close to similar, and we may just be having trouble finding some of them in dog. Um, so back before Tasha was sequenced, the way people went at sequencing was basically using gels, right? And you can have an army of grad students and still not get a whole genome that way. Also, it was radioactive, so that's not so much fun. But by the time that we were starting to sequence Tasha, we had Sanger sequencing, um, costing $3 billion to sequence the first human genome. Tasha came in, or she was sequenced around the time that we still had Sanger. And then when next generation sequencing came around in 2007, that was also part of sequencing Tasha. After that, cost per genome was dropping to 5 to 10,000. And these days, at least at the Broad, we're paying $750 for a 30x genome. This is a picture of possibly one of the most resequenced dogs in the world. That's Besco. It's my PI's sister's dog, who we basically use whenever we need to test out new sequencing technology. We do Besco again so that we can compare to all of her previous sequences. So Besco is, her genome is very well described. So in other words, we've gotten pretty good at sequencing genomes, and it's getting cheaper and cheaper. And there are a whole lot of different groups out there, a lot of different initiatives, sequencing a lot of humans. There's more and more dog sequencing starting. A couple years ago, Elaine Ostrander's lab released 435 complete dog genomes. I wanted to update that on this slide. Carlson Lab has released, I want to say it's just under 700 since then, which includes these 435 and a few more. And these are all deep sequence, so they're all around 30x. But we don't have the URL for that. It's not like fully released yet. So I figured I should stick with the old one that was released. But the problem, of course, is, again, as you all know, is you can have the sequence but you don't know what it means. And you can find the locations of the genes, but again, that doesn't necessarily mean that you know what they are doing. You can try to find regions that you think are important regulatory regions, but we still don't know what those are doing. And then there's the massive amount of the rest of the genome that we just have no clue still sort of what it's there for. And we know that the regulatory sequences tend to be the ones that control behavioral traits. It's less common with behavioral traits that you get an actual functional mutation. They tend to be controlled by a large number of genes, and it tends to be more the regulatory sequences that are controlling them. So that makes it harder, right? It doesn't mean that you can find a mutation and go look at it and see that it's obvious that it has changed the protein, and then that will zero you in on that this is one that is important to chase down. Regulatory mutations are a lot harder to find. Um, and of course, you'd only need a very small number of differences between two dogs to have some really literally enormous changes between the two dogs. So what we do is an approach called GWAS, which many of you probably already understand pretty well, where you look across the genome and you try to look at each location where there is a mutation or at some a marker of a difference and say, is this correlated with the trait of interest? The first way that Carlson Lab, I think this was even before it was Carlson Lab, did this was with coat color in boxers, which we figured was going to be fairly easy to find. It's at least much easier to describe than a behavioral trait. So looking at boxers coming in either solid brown or solid white, or what's called flash, which is mostly brown with some white. And it was known just from doing ancestry studies that this was pretty much Mendelian and that the flash was the heterozygote. Also, interestingly, about 2% of the all white dogs are deaf. So just sort of a side note on that phenotype that becomes interesting later. So again, known that we have the homozygote brown, heterozygote flash, homozygote white, and this initial study was done with, I think they would have preferred to have 10 solid and 10 white, but Tasha, pictured in the middle, is herself. Again, remember Tasha was the dog that the dog reference is done of. So she was used because they had her as the reference and they knew that she was flash, and so that was why they had one heterozygote in there. So for those of you who aren't used to seeing a Manhattan plot, this is the output of the analysis where each different color section here, it represents a chromosome. The little dots represent SNPs, the markers, where one marker is a difference between the two different genomes. And you can see when you take a look at it that, so the markers as they are higher up along the y-axis means that it's a more significant difference. And so just eyeballing this, you can see that there's this one spot on chromosome 20 where there's clearly a really important difference between the phenotype of being brown and the phenotype of being white. And we got lucky in this case that this region was sitting right over a gene, MITF, which was known to control white spotting in other species. So digging down into the story in dogs specifically, turns out that the MITF gene, so here's a picture of the exons are the thick bars and the introns are the lighter blue in between. And there are a couple of different versions of MITF based on which exons are spliced in. And the A version is basically the whole gene. And the start of A then is on the far left. And that version is needed for healthy eyes. And the M version just starts midway down. So if you chop off those first 4 exons, you have the M version. And that's what's needed for normal pigmentation. And the mutation that was found in the regulatory region right there before the beginning of the M version, where that red line is. So what was realized was going on was that the regulation of the M version was disrupted. So just that one shorter protein, the M version, what that protein does is it makes the pigment cells migrate while the dog is still an embryo. So they're migrating from the neural crest. Basically, you can think of it as coming from along the dog's backbone. And those cells, the pigment cells migrate from along the dog's backbone down towards the dog's ventrum, the dog's stomach. And with there's not as much of the protein to help them migrate, then they're not going to migrate as far. So with a brown dog, the cells can migrate all the way around because the protein is being made fully. There's plenty of it, and so you get an entirely brown dog. With heterozygote, there's only half as much of the protein made, and you see that therefore the cells don't always get quite as far. And so this puppy is nicely lying on his back to demonstrate the white spots on his belly and his paws and the line down the center of his face, which are just where the cells, the pigment cells, didn't get all the way there because there was not as much of the protein needed for the cell migration. Obviously, with the white dogs, there just is none of that protein for pigment cell migration, and they are all white. So it's this story sort of highlights why it's useful looking at dogs rather than taking what might be considered a more traditional approach of looking at a mouse. So if you suspected MIDF as the gene that is controlling color, what you might do is a knockout approach in mice. And what you would do then is knock out the entire MIDF protein, not just the M version, but the A version as well. And when you do get white mice. They are also deaf and blind, and they are actually born dead. And so not all that useful for testing, say, behavioral traits, which was where we were going, hard to behaviorally test a dead animal. On the other hand, if you look at these existing populations that are living in people's homes, you can do GWAS to test regulatory variants. And so these dogs are white versus brown, so you can find the variant, but there are still living animals there that sometimes are deaf, but they have normal eyes. So a reason to look at this particular approach. So as people were applying GWAS to dogs in the early 2000s, there were a lot of excellent advances made. So white coat and deafness is what we've just been talking about on the top left. Also the gene for controlling whether a dog is hairless or not, whether the dog, the Rhodesian Ridgeback, what the gene is that controls that ridge along its back, and some other really interesting findings. However, what these things have in common is that they are single gene traits, Mendelian traits, and much easier to find with really small sample sizes. So, gosh, I hope this video comes up for you guys because I know it's probably pretty jerky over the screen sharing. But what Carlson Lab moved on to next was looking at a behavioral trait at OCD. So it's a Doberman Pinscher in the top right and a Bull Terrier in the top left. The Doberman Pinscher is flank sucking. The Bull Terrier is spinning. So these are behaviors that are normal, but are done to really upsetting lengths in these dogs to the point that the dogs can't live normal lives. And they are highly heritable traits. So we knew that they were going to be a good target for doing this kind of work. And they're also begging for more work to be done on them. These are traits that are, when they are pathological, treated with SSRIs, and the response to treatment for them is pretty similar in dogs and in humans. It's just not, SSRIs don't fix it in everybody. They don't even affect it in some people. So there's a desperate need for more medications or more approaches to solving compulsive disorders. I'm trying to get my slide to advance. Great. Yes, that is the next slide. So a glance at this Manhattan plot will show you that whereas the previous Manhattan plot looking at coat color had that one really gorgeous large locus, it's a lot messier here where there's a couple of different spikes. But there is one obvious one, which we did follow up on. We were lucky. This was over a cadherin gene, CDH2. And cadherins, among other things, hold synapses together. So if you imagine that the presynaptic neuron is coming down from the top, postsynaptic neuron is coming up from the bottom, and as these two neurons want to stay together to form a synapse and have a conversation with each other, part of the thing that forms the synapse is this cadherin protein that holds them together. And it's specifically found in glutamatergic synapses, a specific type of synapse. And so we believe that this affects the plasticity of the synapse for learning and memory. And the interesting thing in this study is that the study was done in Doberman pinschers, and it was found that not only was there this one particular difference in dogs that were likely to have developed compulsive disorders versus those that didn't develop compulsive disorders, there's also an area of the genome that was pretty much entirely fixed in Doberman. So it just looks the same in all Dobermans. Um, and this area of the genome contains the gene CTNNA2, which also has to do with anchoring two neurons together in a synapse. It is the protein that actually attaches the CDH2, which had been found to be important in this disease, to the actual neurons. But because it was entirely fixed in Dobermans, we couldn't see that there was any difference in this particular gene between Dobermans that developed compulsive disorders and Dobermans that didn't develop compulsive disorders. We had to go out to look at other breeds of dogs to discover that, in fact, yes, there are risk variants in CTNNA2 for compulsive disorder in other breeds. It appears that Dobermans are, in fact, fixed for the risk variant. So it's part of this story then is that looking at individual breeds of dogs can be very powerful, but can also set you up to miss some really important information. If Dobermans had been the only breed studied here, then the difference in CTNNA2 would have been missed because they don't vary for it. So it was a lovely study. It was a study that got published as sort of one of the first behavioral genetic findings in dogs. But it actually ended up not explaining a large portion of the risk for compulsive disorder in Dobermans. About 10 to 12% of the risk was explained by this particular variant. So what we really wanted was for it to look a lot more like the coat color thing, right? With the homozygous, heterozygous, and then homozygous affected. It was 100% not what we found. The variant definitely affected the risk of OCD and in fact of the severity of the compulsive disorder in the Dobermans. But it was quite possible to be a dog that was homozygous, having the risk allele and being completely unaffected. So, and that is even so a very strong finding, a large effect size for behavioral genomics. But what it did tell us was it pointed us at the importance of glutamatergic synapses in dogs for compulsive disorder. And the other interesting part of that story is that other labs had found that glutamate signaling is also important in compulsive disorders in humans. So in fact, we did go on to publish this paper in Nature Communications in 2017, showing basically this slide shows a picture of a glutamatergic synapse, and it shows all of the proteins that are interacting here. And there's a picture of a human head, a mouse head, or a dog head next to the various proteins implicated by dog, human, or mouse studies. So it's going to show that looking at a particular disease in just one species can get you so far, but looking at that same disease again in different species can start to help you really put together much more of a full picture of what's going on in the disease. And then again, of course, the story continues to be that behavioral traits are very complicated and there are a lot of genes affected in a behavioral trait. And so we in our lab like to show this picture as a way of sort of visualizing what's probably going on. If you think of each of these dots as a gene that affects behavior, and they are all interacting with each other in very complex ways that are very hard for us to understand. And that is sort of what we think the genomic architecture of a complex behavioral trait probably looks like. So in order to get at these very complex traits, we are obviously not going to be able to continue to use a 20 dog study like we did for coat color. And there have been some studies recently on the human side that have gotten um, some really nice findings. So this one looking at schizophrenia, obviously a behavioral trait, found a large number of loci implicated in schizophrenia, but they did it with about 150,000 people. And there's a lot of structure out there for collecting genetic samples from people, but not so much structure out there for collecting genetic samples from dogs. We do hope that behavioral genetics is going to be easier in dogs than in humans, in large part because of these interesting separate populations that we like to call breeds in dogs, but we're still going to need a whole lot of dogs in order to make headway. So that was basically what caused Eleanor Carlson to put together Darwin's Ark, which is this enormous pet citizen science project. Basically, people come to darwinsark.org and sign up their dogs and answer about 200. We like them to answer all 200. They don't always answer all 200 behavioral questions. So this is pretty much what it looks like. We asked, does your dog, how true is this statement for your dog? And they can strongly agree, strongly disagree, or something in between. And at the end of a series of questions, we will give them some feedback on how their dog compares to other dogs. So in this case, at this point, this picture was taken obviously pretty early on. We have a lot more dogs than this now, but showing that most people answering this question said that their dog did enjoy playing with toys. Then when we have the funds for it, we send saliva swabs out to dogs, to the dog owners. For a lot of dogs, it's pretty easy to get the saliva. A lot of dogs will just chew on it, as is pictured here. Although for a lot of dogs, you can just sort of slip the swab in between their teeth and their outer lip and soak up a bunch of saliva. And then the kit is mailed back to us. And there have been a lot of people participating. We are well over 20,000 dogs signed up now. And for individual answers per dog, we have more than 2.2 million answers at this point. And we keep having trouble counting how many DNA samples we have. More than 4,000, probably closer to 5,000 DNA samples, of which we have actually sequenced about 2,000 of them. And the kinds of questions that we address, we address some of the straightforward questions. How much white fur does your dog have? Is one that, it's not a gene that we're going to be super interested in fully mapping out, but it's one of those easier questions to tackle in the time before we have enough dogs to be tackling the behavioral questions. Questions like how long is the dog's fur? What's the dog's ear shape? And how tall is the dog, which we measure by asking how tall is the dog standing next to a human. And then a bunch of behavioral questions. Does your dog retrieve objects? Does your dog point? Does your dog seek companionship from people? And we have gotten some nice distribution of answers. So these are when we do some factor analysis to pull the questions together, the distribution of, for example, questions that seem to relate to impulsivity. We do have a bunch of dogs in the middle and these sort of tapering on the edges. Anxiety, you can see it's not a normal curve. Fewer dogs are anxious than dogs that are anxious. But we have nice distributions, which means that it makes the GOS much easier than if everybody is crowded into the middle. The problem, as I alluded to at the beginning, is being able to afford the sequencing for each dog. And what we're working with right now is low pass sequencing. It gets us close to 9 million SNPs per dog, which is about 12 times as many as when we were using the Affymetrix genotyping array. It costs a lot less than whole genome sequencing. So whole genome sequencing currently $750. Low pass gets you about 1x rather than 30x, but the concordances with the whole genome sequencing are very high. So it's very accurate, and it only requires a teeny tiny amount of DNA, which is really nice because sometimes owners only send you in a fairly small amount of saliva, depending on how easy or not easy it is to collect from the dog. And well, this is a slightly outdated comment that all cheek swabs run so far worked. Actually, some of them have failed at this point, but it has been much more successful than when we were doing the AFI arrays. The AFI arrays were requiring a lot more saliva and a lot higher quality DNA, and therefore were much more likely to fail. So taking a look at dogs that were genotyped on the Affymetrix array, so about 770,000 SNPs, compared to dogs that we sequenced with low coverage sequencing and then imputation to get the variant number up, looking at a GWAS on dog height, how did they compare? So when we did the Affymetrix analysis, we recapitulated a bunch of findings that had previously been found for dog height. We went back and redid it with dogs that we had done low pass sequencing on so that we had many more SNPs. We found some new regions that had been that were interesting because they were over genes that other labs had found related to height in humans and cattle and horses, but that had not previously been found in dogs. Slightly funny story about that CD36 region. It's obviously a pretty high spike, and when we were first looking at it, it um, had not been previously implicated in any studies that we could find about size in dogs. And so partly we were excited that we'd gotten this really strong finding, but partly we were perplexed because it was the highest peak. And so how is it possible that no one else had found it? And literally several hours before I was at the dog and cat conference about to present on it and say, well, we found this cool thing, we finally realized what was going on, which is that this is the gene, this is actually FGF4, it's the retrogene for chondrodysplasia, for short legs. And so all the previous studies for size in dogs had used weight as a way of measuring the dog size. And so obviously when you're measuring weight, you're not going to find short legs are not going to be an important finding in that. But when you are measuring height, short legs are going to be a very important finding in that. So just be careful, I guess, of thinking about how you're measuring what you're looking for, whether what you're measuring is actually what you're looking for, and whether it's the same in the studies that you're comparing to. But luckily, I was caught in time before I made a total fool out of myself with that one. So right, so basically, my summary here is that yes, when we are looking at size, this seems to work pretty well, and even found some new regions that had not been previously published. How does it work when it comes to behavior? You know, we don't have the sample sizes yet to be finding really interesting things about behavior. What we did find is looking at, sorry, I want to see the slide again. Yeah, so looking at how much Labrador Retriever is in a dog versus how interested the dog is in retrieving was an analysis that we were able to do here. So we had, at this point, we had 882 sequenced dogs that had some Labrador Retriever in them. And we were able to say what % lab and then compare to whether the owner asserted that they strongly agreed or strongly disagreed that the dog was interested in retrieving toys. And it does appear that dogs with more Labrador Retriever in them were more likely to be interested in retrieving. So that was an interesting way of getting at behavioral findings, and particularly in leveraging the power of, again, these separate populations, breeds, which are something that dogs offer us that human genomics does not offer us, this having been bred for these particular tasks. So as a sister project to the Darwin's Ark project, we also have the Working Dog Project, where instead of working with owners who have pets living in their houses, we work with assistance dog schools, and we're hoping to expand to other types of working dogs in the future. So so these schools, some of the larger schools are breeding large populations of dogs, several hundred dogs a year, and they are hoping to place most of them as, for example, guide dogs at the guide dog school, but they assess the dogs early on, and then repeatedly throughout their training. And some of them end up continuing on to be guide dogs. Some of them, perhaps if they're higher energy, are going to do better as scent dogs. Some of them might not be quite up for the really demanding task of being a seeing eye dog, but could be assistance dog, perhaps for someone who needs objects picked up for them or something like that. And some others of them go off to the super important job of being companions. And these schools really want to be able to identify which dogs are going to be best for which jobs as early as possible in their lives. They don't want to keep a dog who's destined to be a scent dog and put it through a year of guide dog training before they realize that it's going to wash out. So they do behavioral assessments, but they would love some genetic testing as a way to help them make those choices. And they have been collecting some fabulous data for us. This is, by the way, we were talking before this, I was talking with Matt about whether this was the cutest talk he had seen, and I'm submitting this picture as my submission for the cutest possible picture of a Labrador puppy with a tiny little fake guide dog harness on him. So the guide dog schools are collecting some really cool behavioral phenotypes, such as when a dog is stressed, does he stress down and sort of like, shiver and huddle in place? So they call that inhibited by stress. Or does he start running around barking because he's so stressed? That's activated by stress. Is he sensitive to his harness? Something that they're testing here. Does he have other actual pathologies such as thunderstorm phobia, hip dysplasia? Is he liable to grow up and develop mast cell tumor? So they have all of this great information about their any dog that has passed through their program. So hundreds of dogs over the last decades, actually. And so we're taking this information and sequencing a lot of their dogs. And what we are trying to find is, obviously we're going to take that information and put it into GWAS for our own benefit, but for their benefit, and the reason that they're so eager to work with us, we are hoping to be able to produce genomic EBVs. Those are estimated breeding values. And genomic estimated breeding values are used to great effect currently in breeding corn, also in breeding cattle. And in cattle, using this technology has increased the milk output incredibly dramatically over the past decade, something that I had to listen to people talking about ad nauseum when I was doing my animal science degree. So basically, you find associations similar to GWAS. You find associations between genetic variants and phenotypes, but you output this much more complex sort of, in some ways of doing this, almost every marker is taken into account so that I can sequence a dog. Once my model is trained, I can put the dog sequence into the model, and the model can come back saying, yeah, this dog is likely to have a genetic predisposition to develop this trait, in the positive or the negative direction. Obviously, for behavioral traits, environment is a very important part of the measurement, and the guide dog schools know this. But they also like having this additional piece of information to take into account alongside of their behavioral testing. And they say they might be able to do things like, if it comes back that this is a dog that seems to be genetically at higher risk of developing anxiety, they might then, when they have it as an 8-week-old puppy, be more likely to place it in one of their much more experienced puppy raising homes rather than a brand new puppy raising home. So they could make some of those decisions. So that's what we're hoping to develop there. So the stuff that I'm working on currently, this brand new version of what we call CanFam, the dog reference, has come out. In fact, there are several competing versions. It's kind of hilarious. It makes some of the other computational biologists want to rip their hair out. I just think it's really funny. So there's a new version of Tasha coming out. I think that one hasn't actually been published yet. There are 2 separate German Shepherd dog references coming out. So as after we determine which one we're going with, we need to realign all of our 700 or more deep sequence dogs to this new reference, which has a lot of gaps closed, and also apparently the original reference was missing a lot of first exons. So we're going to get those back in this new reference, which we're all very excited about. And part of why this is an interesting project is it's not just straight up aligning, but I get to learn how to use the Google Cloud platform, Google Computing platform, because trying to run this many alignments over this much data just on our regular computing platform, even as powerful as it is at the Broad, the last time we did it, it took, I think, 6 months. And so I get to learn to use the cloud, cloud computing, which will be fun. And I mentioned that we've been doing low pass sequencing with imputation. So for those who don't know what that means, basically low pass sequencing gets very low coverage. So where we would love to have 30x coverage on all of our dogs, low pass gets us 1x, which is still more markers than a microarray, but not as many as we would like ideally. So then we use imputation. So we have this panel of, again, about 700 dogs, and we use that to impute. So basically that means that if you know this marker and then over there that marker in this dog, you can go look at the panel of the deep sequence dogs and say, well, I know this marker and I know that marker, and then based on linkage disequilibrium, I can guess with pretty high accuracy what the markers that we know of in between those two markers will be. And so using imputation, then we can get up to about 9 million markers per dog. The unfortunate part of that as compared to a microarray is that with a microarray, you know exactly which markers you're going to get, right? That's how a microarray works. And so you can, as for example, Embark has done, put some additional stuff onto the Illumina chip to say, we're really interested in looking at these particular markers, and we want to make sure that we get them every time we genotype a dog. So for example, if you want to be able to tell someone, come and test your dog with us, we will let you know about the answers to some health associated markers that you might be interested in. You can guarantee that you'll get those if you're using a microarray, but not if you're doing low pass sequencing. So the solution to that is targeted sequencing, which is basically a technology where when you do your low pass sequencing, so far as I understand it, you basically put some primers in as well in order to focus the sequencing on the particular areas of interest. So we are currently working with GenCove, the company that we are doing low pass sequencing with, to develop a targeted sequencing panel for dogs. So I have about 200 regions, and some of those are SNPs, and some of those are larger regions to cover like signs and things like that. And so we are with them developing this panel, and when that panel is complete, it will be the same financial cost to sequence a dog as it has been, but the markers will be distributed more over the regions that we have a particular interest in, so that we'll be able to guarantee that we can get back the particular loci that we really want to have back. And part of that is so that we can work with these guide dog schools and say, don't go spend hundreds of dollars paying for genetic testing elsewhere. Come do your genetic health testing with us, and for the same cost. And at the same time, then we'll be able to use that data for research. And then the thing that I'm most excited about is with all of these deep sequence dogs, I'm taking a structural variant package developed at the Broad for use in humans, and using it and modifying it so we can call structural variants in dogs. And I think probably a lot of you are familiar with the story, common in genomics, where we tell the joke about the guy who lost his keys in the park, but he's looking elsewhere because this is where the light is. And of course, that is what we do in genomics. We look at SNPs because SNPs are really easy with microarrays and with sequencing. It's really easy to call SNPs. But we don't actually think that most of the variation we certainly don't think that all of the interesting variation lies in SNPs. And in fact, in dogs, there has been previously published some really interesting stories about copy number variation. As Kelsey surely knows, there was a really interesting paper that came out like 2013 or so, was some of the first really deep sequenced dogs versus wolves studies where they found that one of the big differences between dogs and wolves, and therefore we think a marker of domestication, is the copy number of a gene for digesting amylase, meaning that dogs had evolved to be able to digest starch and carbohydrates, presumably because we as humans were producing those and offering them to dogs in a way that wolves had not evolved to do. And by the way, follow-up papers found that breeds that are up in northern climates, huskies, had had fewer copies of this gene, just like wolves, presumably because up there they were mostly being fed meat. So we know that copy number variation is a very interesting place to look. We also know that signs contribute to a lot of interesting variation in dogs. For example, that beautiful merle coat color that some of you may know of, so common in Australian Shepherds, where the dog is sort of this light gray color mixed with black and then generally has white markings. That is caused by a sign dropping into the middle of a coat color gene. So we know that there's plenty of really interesting information there with transposons, but there's not an easy way to call those right now. So what we're hoping to do is to build up a panel of locations of different structural variants and turn that into an imputation panel such that I would be able to do low pass sequencing, get a bunch of SNPs, and then basically use the SNPs to impute with high accuracy the structural variants. So I'm doing a lot of the computational part of the computational biology, trying to get that pipeline running with dog. It's always interesting trying to get a human pipeline to run with dog. There's always the issue of different numbers of chromosomes. What I'm struggling with right now is that chromosome Y is not characterized currently in the dog reference. And so I'm trying to explain to the software that the way you tell the sexes apart is either it has two X's or it has one X, but don't go looking for the Y because that's going to confuse you. And then that has gotten me into a lot of learning about basically how cloud computing works. It's just the wave of the future, but it's a really different way of doing this computational stuff. So learning a lot about Docker and GCP and things like that. So that is what I have for you. I'm hoping that you guys have questions for me, and I will leave this on this slide, which has my email address. I am always really happy to take questions by email. And I also want to sort of give a shout out to very little of this is actually my work, as you could hear at the end, is basically about what I'm working on. Some fantastic work done by all of my colleagues here. So, all right, tell me you have questions.
Thanks for sharing your research with us, Jessica. Does anyone have any questions?
Matt responded with a clap emoji. That's cool. I haven't seen anyone manage to do that before. That was very nice.
I actually have a question for you, Jessica.
As the moderator should, if no other questions are forthcoming. What is it, Kelsey?
Has there been much work to look at in mixed breed dogs, how the different breeds that make up the mixed breed, how that ancestry is distributed? Is it like in big chunks, or is it kind of really broken up?
Oh yeah. Right. So actually, we are publishing what we've been calling the MUT paper, hopefully soon. We had a meeting today where we're like trying once again to outline the paper. It's like semi-written, but we're going back to the outline phase. You know how that is. So hopefully we'll be publishing that in the next few months, although we've been saying that for a while. And that will cover some of that. Yeah, it turns out there's a lot of dogs out there that are super mixed up. And when you try to do what we call chromosome painting, so that you look at the chromosome and you can see the bits of different ancestry on the chromosome, they are, yeah, it can be very, very mixed up. It turns out to be challenging to do the chromosome painting well because you have to reliably phase the chromosomes. And in order to do that, you have to have pretty deep coverage. Longer reads would be really nice too. And so with the coverage that we have, we've sort of been going back and forth and like, we can kind of do it, but we're not really like confident enough about it to like hand it back to someone, like someone who has their dog sequenced by us. We don't want to give them a picture and say, we're confident that, you know, this is here and that is there. But we're working on it, and we do have some really interesting pictures of some very mixed up dog genomes. If you remind me later, I can send you Jenny's picture from Darwin's Ark, where it's like lots of little bits of things, and then the whole chunk of like, we have no idea what this is. So, which also, there's a lot of dogs out there that, so we are able, with our number of markers, we can call breed chunks with accuracy if the dog is at least, I think, 5% that breed. And there's a lot of dogs out there, like Jenny, I think 30% of her genome we couldn't call, which strongly suggests that what's going on is that 30% of her genome is made up of bits of breeds that she is less than 5% of. So there's a lot of dogs out there like that. I mean, it's also possible that those bits are like breeds that we don't have in our database, but we have all the most common breeds. So I think that's pretty rare. And then there's the question of whether there are dogs out there that are not any breed, like, are there dogs that don't have European breed origin in them? And so that's sort of an interesting question too, that we're still figuring out how to go forward with.
Cool. Any other questions?
I want to hear from Matt if I win with the cutest picture. He's like, no, I can't figure out where the unmute is.
We'll say you win by default then.
Okay, thank you. Well, I know a lot of people were going to a different talk, so if they have a question, they should feel free to email me.
Yeah. Well, thanks everyone for coming, and thanks again, Jessica, for sharing your work about dogs. It's always exciting.
Thank you. Thanks very much for having me. I really wish I could have come down to see you guys in Providence. I love Providence.
Oh, I know.
Some other time.
Hopefully soon.
Yeah.
But take care, everyone.
Thank you, Jessica.
Bye. Thank you.